So question number 10 was really tricky, so it's worth just going through this text really carefully. So scientists are investigating the function of a eukaryotic cell protein called cyclin A. So I'm just going to pop that info here to remind me cyclin A is a protein. This protein is thought to be involved with the binding of one of the enzymes required at the start of DNA replication. So it's a protein that we think is required to bind a second protein at the start of DNA replication. And then we've got 4 conditions. So we've got control cells untreated. We've got an antibody that binds to cyclin A. So in D, we're saying an antibody binds. Now, we haven't really looked at bodies again, but think back to GCSE. Antibodies, we know, bind to pathogens and destroy them. It's going to have a specific shape. Maybe that binding is going to block this binding. Okay, so that's happening there. E, they added RNA that prevents translation. So I'm going to go to another colour here and saying, okay, cyclin A is a protein. So that means in order for cyclin A to be present, we have to have some translation of mRNA. We have to have transcription of the DNA. So there must be a gene for cyclin A, which is translated, transcribed to make cyclin A. And in E, we're saying that they're adding RNA. That's not really relevant, a molecule that prevents translation of cyclin A. So I've got mRNA for cyclin A present, but I haven't got the protein being made. And finally, in F, they are doing the same. Okay, so in E and F, they're doing this, but they're also adding more cyclin B artificially. So going through here, we're saying in the control, cyclin A is present in whatever its normal amounts are. We don't know what those are. In D, cyclin A is present, but cannot bind to DNA because it's bound to an antibody. In E, we're saying that there will be no cyclin B or cyclin A or reduced cyclin A because there's no translation of protein, so the protein can't be present. And in F, we're saying there is no cyclin A being produced, but we've added more artificially. Okay, so that's what I'm taking just from this question before I've even looked at what I'm going to get asked about this. And it's really useful when you look at these questions, especially these complicated ones where you've got 4 conditions and 4 sets of results, that you start to think this way, okay, what's going on in each one of these? Don't just read through it and go straight to the question. You've got to take that time, especially in these end of topic tests where perhaps you have a little bit more time than you will in the real A level, so you can get into good habits, and then in 2 years' time, you'll be much quicker at this. Okay, so I'm going to look at this question, and the first thing I'm noticing is that it's a suggest question. You'll remember this from GCSE. Suggest means we haven't studied this, so we haven't studied cyclin A or its role, but we just have to look at the mechanisms and think what explanations could be possible here. Now, looking back at that data, it's a 3 marker, and I've got a control plus 3 conditions. So if I work through each one of those conditions, then I should pick up the marks. Okay, so a mistake that was made here was lots of people just repeated the data back at me. That's a description. Okay, so description might be tell me what you see, analyze might be do a calculation, but for these explanations, we need to make an observation, but then give an explanation, and therefore is a really useful word to use for that. So in D, we're saying that the antibody binds to cyclin A. You don't get any marks for that because that's in the box. But then I've got to say why. So therefore helps remind me that I need to explain this. If that antibody is binding to cyclin A, that means that cyclin A cannot then bind to DNA because it's got a protein bound to it. And we know from our study of things like enzymes that a protein binding to another protein could alter the shape of that protein, and therefore cyclin A can no longer bind to the DNA. So my explanation is telling me why that antibody binding to cyclin A has reduced that DNA replication so much. If I move on to E, we've got a similar reduction. So then I'm going to try and explain E. Now, we don't have to know how that RNA prevents translation of cyclin A. We just need to make some suggestions based on what we understand about translation. So how is it going to do that? I'm going to use therefore again to remind me that I'm explaining prevents translation of cyclin A. Therefore, this RNA must be somehow interfering with the translation mechanisms. So I can say RNA interferes with the mRNA or with the tRNA, or maybe it interferes with the ribosomes, or maybe it interferes with that polypeptide formation. So we don't know. The actual answer is beyond A level, and actually that RNA is probably interfering with the mRNA. But the point is to make the suggestion based on what you guys know about translation, which is that translation involves these bits of mechanism, mRNA, tRNA, ribosomes, and polypeptide. So your explanation needs to mention those. Finally, treatment F shows us something slightly different again. So in F, we've got those two molecules added. We've got something preventing translation of cyclin A, but we've also artificially added back in more cyclin A. So we've got added cyclin A, which shows that the other treatments are being affected by this loss of cyclin A. So therefore, if adding cyclin A restores this function of DNA replication, therefore cyclin A is affected in the other treatments and is also required for DNA replication, which is only a suggestion in your stimulus material. What do I mean by that? Well, if we just scroll up quickly, then we can see up here it says scientists investigated the function of cyclin A. This protein is thought to be involved. So it's thought to be involved. F is the one that helps us to prove that it must be involved. So really tricky question. My top tips are just look at that table really carefully, keep referring back to it, use therefore constantly, make an observation, therefore. The answers I read, lots of people made a really good effort, but ultimately were just describing the results to me over again, rather than trying to explain them.